These pages start with a summary section, which gives you an overview of the information that we have, the structural information that we have for a certain protein. For example, in this case, what you can see is we have a box which gives you textual summary of this protein. You see the name of the protein, you see the gene name, and also flags for enzymes. And in fact, these are links to Brenda, so you can learn more about the specific enzymatic activity that this protein performs. And also, if this protein is known to be implicated in disease, then we have detailed information on that too. We also have description of the biological function, and this actually is coming from UniProt. Then on the right-hand side, we try to give you an overview of all the available PDB structures for this full-length protein. So I will go into a bit more details regarding this component. At the top of this visualization component, you have a help text tooltip. So if you were to go there, you would see a brief description of what we have available within this component and what is it that you see here. Underneath, at the bottom of this component, you can see a link which goes directly to the chain that is being displayed here. And under that, you get information regarding the coverage, which basically means that this particular chain, for which you see the structure here, is found at these residue ranges in terms of the UniProt sequence. And this actually is 10% of the full-length sequence of UniProt. There are several ways, or a couple of ways, to navigate between the different PDB entries mapped to this UniProt sequence. One is to use these arrows, and the other is to use these boxes. Each of these boxes represent a specific PDB chain. The currently selected one is always blue, and if you click on another one, then it will turn to that structure and show it there as well. If you click on the image itself, then it will link to a 3D visualization tool, LightMole. And this is an interactive tool where you can get into the details of the structure itself. Finally, at the summary section, we have ways to navigate between the different parts of the page. And at the top, we have a navigation bar. And also we have these icons, which on one hand serve as navigation. So these are buttons that you can click on. And also they give you an overview of the available data. So for example, in this case, what you can see is that for the crab binding protein, we have 88 structures, 81 unique ligands observed in PDB. This protein is found in 5 interactions with other proteins or nucleic acids. We have 3 additional functional annotations, which I will detail later. And we have almost 400 publications. So this covers the summary section of the page.